RNA transcription is like creating a blueprint for a house. During transcription, DNA is used to create an RNA sequence that will later be used to create proteins. RNA can be represented as putting plans for a house onto a blueprint for the builders to use. RNA polymerase is the main architect in the RNA construction process in prokaryotes. RNA polymerase makes phosphodiester bonds to add RNTPs to corresponding DNA. RNA polymerase is made up of many different subunits that together form the core enzyme. The sigma factor is an important subunit that recognizes the promoter region on the DNA template. Once the sigma factor binds to the core enzyme, it forms what is known as the holoenzyme. Our promoter region contains what is known as the -35 and -10 consensus sequences. Consensus sequences are sequences that contain nucleotides commonly found in a region. Usually in bacteria, the -35 sequence is TTGACA, and the -10 sequence is TATAAT. These sequences are named -35 and -10 because they are 35 and 10 nucleotides upstream of the +1 region, or the first nucleotide transcribed. Like an architect using an ID to get into his office, the sigma factor region on RNA polymerase recognizes these sequences and binds to the promoter, forming a transcription bubble. After RNA polymerase is bound and transcription is initiated, the sigma factor dissociates from RNA polymerase and transcription continues in a process known as elongation. This is where the bulk of the blueprint is made. RNA is added in the 5' to 3' direction. RNA polymerase reads the template strand of DNA to add complementary ribonucleotides. The other strand of DNA, the coding strand, is not read and has a sequence that will match the newly synthesized RNA, except for thymine, which is replaced by uracil in RNA. There are multiple ways to terminate transcription in prokaryotes. They usually involve termination hairpins, which is RNA that has bonded with itself after reading an inverted repeat on DNA. Hairpins often look like this. One way of termination is known as Rho-dependent termination. Rho, a protein, binds to the newly synthesized RNA and moves towards the 3' end. When RNA polymerase reaches a termination hairpin, it pauses. Rho then acts like a helicase enzyme, causing the DNA and RNA to unwind and end transcription. Rho-independent termination does not involve Rho. Instead, RNA polymerase pauses at a hairpin. Following this inverted repeat on DNA, there is a large series of A's, which are synthesized as U's on RNA. A-U bonds are relatively weak, and a long series of them allows for DNA and RNA to separate, ending transcription. What happens if we have an insertion of 5 additional base pairs between the -35 and -10 region that codes for an enzyme that produces the amino acid phenylalanine? Because transcription begins at the +1 site, the gene for this enzyme would not be transcribed and would not be included in the blueprint. This means the organism could not produce phenylalanine.